I distribute the poster, so anyone who interested to listen to your lecture can join. I hope it's okay.
But I don't know, maybe it's good or not good.
It's good. It's good. Thank you, Budina. So, yeah. So, good morning, everyone. So, welcome to the second lecture in this semester with Dr. Jongkon Saising, or we can just call her Bu Aung later. Before starting the lecture, I would like to introduce or read the CV of the speaker, Dr. Jongkon Saising. So, she's Dr. Jongkon Saising. Is currently a lecturer in the School of Health Science in Mapalong University, Thailand. She did her PhD in microbiology in Prince of Songkla University, Thailand, and she also did the postdoc in University of Tübingen, Germany for, I don't know, 1 year, right?
Yeah.
Yeah, and she also published many very good publications. You can see in her profile in Google Scholar later. So I will just directly give the time to Aum or Dr. Saising to start the lecture. Please, Aum, you can share your PowerPoint if you want.
So everyone can see my slide, right?
Yes.
Okay. And good morning, everyone, and every student. I think we already met last week, and we talked about the microbial control, about the many kind of methods, and also many kind of the agents. For today, I will talk to the microbial control tool, which is especially to antibiotics and also antibiotic resistance. Before I start our lecture, maybe I will give you the definition of some key terms of the word that involved in antibiotic and antibiotic resistance. And as you can see here, there are many words, and especially there are very similar. The first word is the antibiotic. The antibiotic is a class of drugs used to treat bacterial infections. And the second one, the antibacterial, actually is very similar. It is used to be a synonym of antibiotics. And nowadays, the substances used to disinfect non-living surface are known as antibacterials. And here the antifungal, antiviral is the class of the drugs used to treat fungal and viral infections respectively. And when we talk about antimicrobial, the antimicrobial is the whole umbrella, the whole term of antibiotics, antibacterial, antifungal, and antiviral agents. And if you look at this figure, for this, the right figure is a very famous scientist. Here is Alexander Fleming. He is the one who discovered the first antibiotic called penicillin. And actually at that time, he just would like to grow the bacteria named Staphylococcus aureus. And after they grow the Staphylococcus aureus on the plate, and he accidentally opened the plate, and that plate, that Staphylococcus aureus plate, is contaminated with the fungi or the mold called Penicillium nonatum. And then after he noticed on the plate and he saw the clear zone or the inhibition zone on the Staphylococcus aureus. He mean surround the fungal, the Penicillium nonatum colony, and there was no growth of Staphylococcus aureus, and that even is a discovery of the antibiotic called penicillin, which is from the mold Penicillium nonatum. And we talk about the antibiotic. There are two types of the activity of antibiotic. We call broad spectrum and narrow spectrum activity of antibiotic. The broad spectrum of the antibiotic, the activity, it means the antibiotic is effective against wide range of infectious microorganisms. For example, they have the activity both gram-positive and gram-negative bacteria. For example, the antibiotic in the group of aminoglycosides and tetracycline. But if those antibiotics which have effective against specific family of the bacteria. For example, the antibiotic called penicillin or erythromycin, they have activity against only gram-positive bacteria. And in the case of methicillin or oxacillin, they both are effective against bacteria in the genus of Staphylococcus. With this kind of activity, we call narrow spectrum. Narrow spectrum only specific some group of the bacteria, but broad spectrum activity of antibiotic specific wide range, both positive or gram-negative, depend on the antibiotic. And a kind of an activity, we call these two kind of activities, we call bacteriostatic activity and bactericidal activity. If the antibiotic, for example, this kind of antibiotic only can inhibit the growth of the bacteria, but they do not kill the microorganisms. We call this kind as bacteriostatic activity. They just only inhibit, but not kill the microorganisms. But if the antibiotic, like in the examples of this group of the examples, if they kill the target microorganisms, they show bactericidal activity. Cidal means kill. Bactericidal activity means the antibiotic kill the target microorganisms. But if bacteriostatic activity, they inhibit but do not kill the microorganisms. And many antibiotics, have their own mechanism of actions. The mechanism of actions of the antibiotic target the cell, target the bacterial cells. And first you have to imagine the structure of the bacterial cells. As we all know that the bacterial cells have the cell wall, and next to the cell wall is the cell membrane or cytoplasmic membrane, and inside the cell they have the ribosome, and they have DNA and also RNA. The antibiotics target these structures depend on the type of antibiotic. For example, the antibiotic that target the cell wall synthesis. These are the example. For example, the antibiotic in the group of beta-lactams and glycopeptides. And apart from the cell wall, the antibiotics also affect the cell membrane, of the bacteria. For example, polymyxins, and they also inhibit the replication of DNA. For example, in the group of quinolones, and for rifamycin, they affect the transcription of the RNA. They affect RNA transcription, and the antibiotics also can inhibit the synthesis of the protein. They act as 50S ribosome inhibitors or 30S ribosome inhibitors in different group of these antibiotics. And I will come to the first mode of action of antibiotics. The first group is the antibiotics that inhibit the cell wall synthesis. And normally, if you remember the structure of the bacterial cell wall, the structure of bacterial cell wall it is composed of peptidoglycan. And with this structure, the bacterial cell can maintain the shape and the size. Whenever the bacterial cell wall is injured, or the cell wall formation is inhibited, and then lead to the lysis of the cell. I will give you the example of antibiotic that inhibit the cell wall synthesis. The first example is the antibiotic in the group of beta-lactam antibiotics. And this is the structure of beta-lactam antibiotics. This is the beta-lactam ring of the drug in this group. This beta-lactam bind to the enzyme of the bacteria. This enzyme is called penicillin binding protein or PBP. With this antibiotic, they inhibit the transpeptidation, inhibit the synthesis of the cell wall, and then inhibit the cell wall synthesis. And this figure will show you the mechanism of the antibiotic in the group of beta-lactam. The green one here is penicillin binding protein, which is the enzyme that link the structure of the cell wall. And then when the bacterial cell exposed to the antibiotic in the group of beta-lactam here, the beta-lactam will bind to penicillin binding protein, and then the structure of the cell wall cannot be synthesized both in gram-negative and also in gram-positive bacteria. And then when the structure of the cell wall cannot be synthesized and lead to the lysis of the cell and the cell death finally. So this is the mode of action of the antibiotic in the group of beta-lactam. And this is the example of antibiotic in the group of beta-lactam. And this antibiotic is, I think you already know, the penicillin, and also the next generation of the group of beta-lactam antibiotics. The second mode of action of antibiotic is the inhibition of cell membrane function. The cell membrane or cytoplasmic membrane or plasma membrane, actually the function of cytoplasmic membrane is the selective permeability barrier of the substance, and the cytoplasmic membrane also carries out the active transport functions and controls the internal composition of the materials within the bacterial cell. Whenever the cell membrane is disrupted, and then lead to the macromolecules and ions inside the cells is escape from the cell and lead to the cell damage or cell death finally. The example of the antibiotic that target the cytoplasmic membrane is polymyxin B and colistin. The polymyxin B interact with the phospholipids in the cell membrane and increase the permeability that can the antibiotic can cross and go inside the bacterial cell and lead to the leakage of the macromolecules and ions inside the cell. And this figure show the mechanism of polymyxin B here. This is the polymyxin B, and this is the here is the outer layer, outer membrane of the bacteria in this layer, and this is the cell wall, the peptidoglycan, and the inner membrane. The polymyxin B here bind to the lipid A, which is the one of the structure of lipopolysaccharide or the outer membrane of the bacterial cell, and bind to the lipid A, and some of the polymyxin B cross the outer membrane and bind to the inner membrane of the bacteria, and then lead to the leakage of the ion which and macromolecules and the molecules inside the cell, and finally the bacterial cell death. The third mode of action of the bacteria is inhibition of protein synthesis. Everybody know the ribosome is the organelles that involved in the synthesis of the protein in the cell. The bacterial ribosome is 70S ribosome, which is composed of the small subunit, the 30S ribosomal subunit or small subunit, and the large subunit, the 50S ribosomal subunit or the large subunit. The protein synthesis inhibitors, I mean the antibiotic that inhibit the synthesis of protein can bind to either 30S or small subunit or 50S or large subunit of the ribosome. When the antibiotic bind to the ribosome and then they cannot produce or synthesis the protein. The example of the antibiotic that inhibit the synthesis of the protein is the antibiotic in the group of aminoglycosides. This is the aminoglycoside and bind to 30S small subunit of the ribosome. And then the protein that can synthesis here is misfoprotein. And when this misfoprotein cooperate to the inner membrane of the bacteria and increase to the aminoglycoside uptake, and then with this misfoprotein and the bacterial cell will die. Finally, the cell death. And this is the examples. These are the examples of antibiotics that bind to 30S subunit or small subunit of the ribosome in the group of aminoglycoside. For example, the antibiotic called gentamicin and the group of tetracyclines. For example, tetracycline, minocycline, and doxycycline. These are the group of antibiotics that bind to small subunit and then inhibit the synthesis of the protein. And while in the antibiotics that bind to 30S subunit or large subunit ribosome are the group of macrolides. For example, the antibiotic called erythromycin and the group of lincosamide, for example, clindamycin, and also chloramphenicol and oxazolidinones that bind to a large subunit of ribosome and inhibit of protein synthesis. And the next target of antibiotics is the inhibition of nucleic acid synthesis. The first nucleic acid synthesis in inhibition is inhibition of DNA synthesis, because as we know, the nucleic acid is DNA and RNA, and the first group is inhibition of DNA synthesis. This group of antibiotics, the quinolones group, for example, ciprofloxacin, ofloxacin, or norfloxacin. This group of antibiotic block the DNA gyrase and then inhibit the synthesis of DNA in the cell. This is the DNA gyrase or topoisomerase enzyme, and this is the antibiotic in the group of quinolones. And when these quinolones bind to topoisomerase, and then the synthesis of DNA is blocked, and when the cell cannot synthesize the DNA, and then cause to the cell death. The next one is the inhibition of RNA synthesis. The inhibition of RNA synthesis is happen when the rifampin or rifampicin bind to the enzyme called DNA dependent RNA polymerase enzyme. Actually, the first figure in the figure is the normal normal elongation of the RNA, the transcription from the DNA strand, and then when the antibiotic called rifampin bind to the beta subunit of this enzyme here. They bind. This is the antibiotic and bind to beta subunit of this enzyme, and then the mRNA elongation is blocked by this binding of rifampin to the DNA-dependent RNA polymerase enzyme. The last mode of action that I will show you today is the inhibition of metabolic pathway. The example is the folic acid pathway of the bacteria. Two antibiotics involved in disrupt of bacterial folic acid pathway. The first one is sulfonamide. The sulfonamide bind to the enzyme dihydroxyapatite synthesis, and then when this antibiotic bind to this enzyme and the para-aminobenzoic acid, which is the substrate in this pathway, cannot be changed to dihydroxyapatite acid. So this is the first step of inhibition of folic acid pathway by antibiotic called sulfonamide. And another step of the inhibition of folic acid pathway by the antibiotic called trimethoprim is here in this step. The trimethoprim inhibits the enzyme called dihydrofolate reductase here, and then cannot change the dihydrofolic acid to tetrahydrofolic acid, the product of the bacterial folic acid pathway. And when we use the antibiotic to inhibit or kill the bacteria, at the same time the bacteria also develop the resistance to the antibiotics, and this is the timeline of the antibiotic resistance, the time life of the use of antibiotic and the beginning of the resistance to that antibiotic. For example, the first antibiotic penicillin here, this it was discovered in 1930s, and then I think around 10 years later, the antibiotic resistance bacteria, the bacteria that resist to penicillin developed in almost 10 years later. And as if you can see here, and for example, erythromycin here, they develop here the antibiotic, and the antibiotic can develop maybe 5 years later. So even we have many kinds of antibiotics that the human had discovered to inhibit and to kill the bacteria, but the bacteria themselves also have the mechanisms to resist to those antibiotics. And the bacteria has many many mechanisms to resist to the antibiotics, for example, and this is the cell of the bacteria, and the bacteria have many mechanisms to resist to the antibiotics. For example, they decrease the uptake of the drug into the cell or inside the cell, and if the drug can go inside the cell to the target cell of the bacteria can pump out the drug, out from the cell. We call the efflux pumps. Or the bacteria can produce the enzyme. We call inactivating enzyme. And this enzyme can destroy the drug, or can cleave the drug. Or they have the alternative enzyme to change the drug from A to B, and even they can alter the target of the drug, and then the drug cannot kill or inhibit the growth of the bacteria. The first mechanism of antibiotic resistance is the decrease the outer membrane permeability, or decrease the uptake of the drug. This resistant mechanism, the decreased outer membrane permeability, can be found in the gram-negative bacteria, and the drug can cause this resistance caused by the lipopolysaccharide composition. The lipopolysaccharide composition of the membrane and the porin, and also the porin protein of the outer membrane, and then lead to the decreased penetration of antibiotics to go into the cell. The next resistance mechanism is the increased export, or the efflux pumps. Whenever the bacteria, the antibiotics can go inside the cell, the bacterial cells have the transporters, or we can call efflux pumps, to pump the drugs out, and then when the cells pump the drug out, the drugs cannot damage or kill the bacterial cells. This mechanism can be found in both gram-negative and gram-positive bacteria. This figure just show you the example of the many family of the efflux pumps. There are family of the pumps that can pump the drugs out from the bacterial cell. And I don't want to go in the deep detail of this figure, just to show you that actually there is only one family of the pump in the bacterial cell. There are many types of the pumps to pump the drugs out. The next mechanism of the bacteria to resist to the antibiotics is the drug in activating enzyme. It means the enzyme that can inactivate the drug. This mechanism can be found in both gram-negative and gram-positive bacteria. The bacteria can produce the enzymes that destroy the drugs. For example, the enzyme beta-lactamase, the bacteria produce the enzyme called beta-lactamase, and this enzyme destroy the beta-lactam antibiotic. For example, here, this is the beta-lactam antibiotic, the penicillin, and the bacteria can produce the enzyme called penicillinase. This enzyme, this penicillinase enzyme, destroy the penicillin when the enzyme cleave the drug penicillin, and the product is penicilloic acid that cannot affect the bacterial cell, and the bacteria also produce the enzyme called cephalosporinase, and this enzyme will destroy the drug called cephalosporin, and this drug cannot damage the bacterial cell. And this is the example of drug inactivating enzyme. This is the beta-lactamase. This is the target of this enzyme. The enzyme esterase or phosphotransferase, and the target of the enzyme. The concept is the bacteria produce enzyme. This enzyme destroyed the drug, and the drug cannot affect or cannot kill the bacteria. So bacteria can resist to antibiotics or the drug. The last resistant mechanism that I will give you the example is target alteration. Normally, the antibiotic, this is the antibiotic. The antibiotic attacks the specific target of the bacteria. This is the antibiotic that target this specific target of the bacteria, the specific receptor. And whenever the bacteria have the mutation, this mutation result in the outer target. When the target of the bacterial cell is changed, this antibiotic cannot bind to the target anymore. Then the bacteria resist to the antibiotic. In the case of the antibiotic, in the case of there is no mutation, the antibiotic can bind to the target, and the bacteria are killed. But when the target is altered or is changed, the antibiotic cannot bind, so the bacteria can resist to the antibiotic. Okay, and this figure, and you can see here when the bacteria or even the fungi they are in many where in our world, and whenever we use the antibiotics can kill these microorganisms which is in the people, in the crops, or in the animals. And then when we use the antibiotics with this microorganism that cause the infection. And as I showed you before, the bacteria themselves also can resist to antibiotic. And these antibiotic resistant microorganisms can multiply themselves to many cells of microorganisms, many resistant microorganisms, and can transfer to this resistance to other cells, and then the antibiotics cannot kill the resistant microorganism anymore. And this resistant microorganism can spread worldwide. And we face the antibiotic resistance microorganism now. How the resistant microorganism can be transferred. They can transfer by the transduction, conjugation, and transformation process. They can transfer the resistance genes by this kind of the process. And how we can prevent the antibiotic resistance. And this is the guideline for antibiotic resistance and what we can do to prevent the antibiotic resistance. For example, we only use the antibiotics when prescribed by the certified health professional, by the specialist, and we always take the full prescription, even if you feel better from the infection, but you need to continue to take the antibiotic after we finish, and never use the leftover antibiotics, and never share the antibiotics with others, and prevent the infections by regularly washing hands, avoid the contact with the sick people, and keep your vaccination up to date. All of them are the example of the prevention that we can do to prevent the antibiotic resistance, which is a very big problem in our world now. Okay, and I think it's done for my lecture today. I welcome any questions from all of the students. Thank you.
Yeah, thank you, Bu Aum, for the very nice and understandable material delivery about the antibiotics, the mode of action, as well as the resistance of the antibiotics. So for the students and for the participants, if you have any question, you can use the feature of the raise hand feature. Yeah. So while waiting, there are two students who raised their hand already. Please, Rita Moses, you can ask your question.
Okay, thank you for the opportunity, sir and ma'am. My name is Rita. I come from Microbiology class C, and my ID number is 079. And here I'm going to ask about the bacterial resistance towards antibiotic. It is said in poster that we always take the full prescription even if we feel better. I don't understand why we must to take that in case to prevent the bacterial resistant to antibiotic. What's the mechanism and correlation between that two topic? Thank you.
Yeah, thank you. Bu Aum, do you want to answer directly the question or accumulate the questions first?
I think maybe one by one.
Yeah, okay.
Okay. From Rita question, I think the first question is why we need to continue to eat or take the antibiotic even we feel better, right? This is your first question.
Yes, ma'am.
Okay, and we need to continue because when we are prescribed with the antibiotic, and the antibiotic can kill the bacteria, but the bacteria not kill all with the antibiotic when you eat for the first time. You need to continue to finish all of antibiotic that you get to completely kill the antibiotic. Otherwise, if you stop taking the antibiotic before they finish, some of the bacteria still alive, and they can develop themselves to resist to the antibiotic.
Yeah, thank you, Pu Aung, for the answer. Do you get the answer, Peter?
Yes, ma'am. Answer very clear. Thank you.
Yeah, you're very welcome. So the next question from Al-Fawzi. Please, you can ask your question.
Okay, thank you for the opportunity. I'm Fauzi Satrio Wijaksono with the ID number 21 from Microbiology A, and I want to ask about the beta-lactam antibiotics, and little they know about beta-lactam antibiotics has a potency, especially the two potency by Woodward's parameter and the Cohen's parameter. And I think why can we decide the best but beta-lactam antibiotics by the Woodward's parameter and the Cohen's parameter? I mean, where is the best one of that two? Thank you.
Okay, your question is, Arif, can you help me, please?
I also still don't get the main question or the main message of the question. Al-Fawzi, could you please repeat slower so we can get your question? Thank you.
I'm so sorry.
Al-Fawzi.
Yeah, it doesn't matter. I mean, either for the potency of the beta-lactam antibiotics, it includes two potency. The first one we know as Woodward's parameter, and the second one it also symbolized as H or H, and the other one is Cohen's parameter or symbolized as C. And how we decide the best antibiotics by that two methods or the two potencies like that. Can you get my question?
The question is about the potency of the antibiotics or the beta-lactam antibiotics, about the Cohen's potential, and what is the other one? I cannot really.
Woodward parameter.
Woodward parameter. So this term were also new for me, the Cohen's parameter and the Woodward parameter. If maybe you can explain what is the Woodward parameters and also the Cohen's parameter at first, so we can understand what you want to ask, maybe.
How far I know about the Woodward parameter is the height of the pyramid formed by the nitrogen atom of the beta-lactamase, and I mean beta-lactam antibiotics, and sometimes called as the base site of the antibiotics. And the second one is the Cohen's parameter, is the distance between the carbon atom and the carboxylate and the oxygen atom of the beta-lactam carbonyl, like that.
Okay, so his question is about the how to choose the best beta-lactam antibiotic based on the structural chemical. I guess that is the main message, because the Cohen and the Woodward parameter is about the nitrogen and also the carbon things in the compound, I guess. Do you have any idea about how to choose based on the compound structure?
Okay, I have no idea about this. Sorry.
Okay, yeah. So maybe if I have if I can give my opinion, maybe it depends also on the strain of the bacteria, right? So every antibiotic or every beta-lactam antibiotic has a certain affinity towards the component of the bacteria. So every antibiotic has a certain activity towards the certain strain. So we cannot really choose one antibiotic for the broad spectrum and bacteria, for example. Depends on the case. I think it's case by case.
Yeah.
Thank you. Yeah. So you get the answer, Al-Fawzi?
Yes, sir.
Yeah, thank you very much. So the next one is Zumrati. You can ask your question, and it would be great if you can turn on your camera.
Okay, thank you, sir, for this opportunity. And I'm sorry I cannot on my camera because the network is bad here. And I'm Zumrati from Microbial GC with ID number 066. And I want to ask questions about the bacterial resistance. How we know that these bacteria are resistant of these antibiotics, and how we solve this problem. Like, should we use another antibiotics that works in different points, or we can use the same antibiotics that work in same point but more advanced? And how we know about this bacteria is resistant of these antibiotics is, should we use the molecular analytics or another procedure like that? Thank you.
Okay. The normal way, I mean, in the daily life, there is the method that can use to test the antibiotic resistance of the bacteria. I mean, this test is available in the lab in the hospital, and this is, I don't know, you can see this one. This is the, how can I say, the routine work in the laboratory in the hospital. For example, when for me, if I'm infected with bacteria, I get bacterial infection, and I go to see the doctor, and then the doctor would like to know the bacteria that cause the infection. For example, I have the wound and the severe wound, and then they take the sample from my wound. For example, the wound, the swab from the wound, or my the pus from the wound, and after the identification of the bacteria, the isolation of the bacteria, they use the bacteria and test for antibiotic resistance. And if you can see here, the bacteria that I isolate from my wound will be cultured on the plate like this. And at the center of the plate is the antibiotic disc. Actually, it is the when they test not only one antibiotic, but they place many types of antibiotic that are available in the hospital. And if the antibiotic can inhibit your bacteria, if the antibiotic can inhibit the bacteria, you can see the clear zone surrounding the bacteria, the colony of the bacteria. But after you see the clear zone, you cannot make the decision that these bacteria resist or susceptible to these antibiotics. You need actually they have the standard table that you can compare the size of the clear zone to the standard and interpret the result into resistant, susceptible, If your interpretation of the result is resistant, it means this bacteria resistant to that antibiotic, and you cannot choose that antibiotic for the treatment. But if from the interpretation, the antibiotic, the bacteria are susceptible to antibiotic, and then you can choose, the doctor can choose that antibiotic for the treatment of the patient. I think for this method, I will show you next week in our lecture. But with this routine method, we can know that the bacteria resist or susceptible to the antibiotic. This is the routine method in the laboratory. And you also use the molecular technique to detect the genes that encode for the resistant mechanism of the bacteria as well. Do I answer your question?
Yeah, thank you for the answer. Very clear.
Yeah, thank you, Aum, for the answer. The next question is from Nur Awalia. Please, you can ask your question.
Yes, thank you for the opportunity. Am I audible?
Yes, we can hear your voice clearly.
Yes. So my name is Nur Awalia Mentari Sukma from class B Microbiology with ID number 89. I want to ask about the antibiotic resistance, that antibiotic resistant germs can multiply. Some resistant germs can also give their resistance directly to other germs. I want to ask about how can the resistance of one resistant bacterium can be transferred to another bacteria, and does it happen naturally or by engineering? I have understand about the resistance from that it derived by their germ, but I don't understand that it can transfer to another bacteria. Thank you.
Okay, you mean the transfer of, I think here the resistant gene can transfer from one bacteria to another bacteria by these three process. And for example, let me see here, the conjugation with this process, it means when you have one bacteria here and you have another bacteria, and this gene from the resistant bacteria can transfer to this bacteria by conjugation. Conjugation is like mating, when two bacteria come close together, and this resistant gene can transfer to another bacteria through the structure called pilus or pili, how we call sex pili. This gene will go to another another bacteria by the sex pilus or sex pili. And for and next example of the transfer is the transduction. The transduction is the transfer of a resistant gene by the virus called bacterial phage. The virus which has the bacteria as their host, and when the virus infect, actually the virus will infect the bacterial cell, and within this process, after virus infects this cell, and the genetic material maybe can exchange with this process. And when this bacteriophage infect another bacterial cell, they will bring this genetic material or this resistant gene to another bacteria. And also with the transformation process, with this process, the bacterial cell can uptake or pick the resistant genes from the resistant bacteria. So the resistant genes or resistant material of resistant bacteria can be transferred to other bacterial cells through these three processes.
Yeah, thank you, Bu Aung, for the answer. Do you get the answer, Nur Amalia?
Yes, I get the point. I want to ask following question about the transformation that the resistant genes that release from nearby life or that can that how that resistant genes can be entered to another bacteria. Is it through diffusion or there is a specific transfer protein like that?
Do you get the question, Aung?
I can get the question, Arif.
She asked about the mechanism, how the transformation works, like whether it's naturally diffused into the cells of the new cells, or is there any specific proteins that help the uptake of the extracellular DNA from the environment?
Oh, actually, and in the environment, I'm not sure, but in the lab, we can make the bacterial cell like ready to uptake the genetic to uptake the resistant genes. For example, we need to prepare. It's like the normal transformation in the lab to prepare the cells to be the competent cell and use many methods. It's like the chemical methods or the physical method, like the high voltage or something like that, to make the pore in the surface of the cell to make the cell to be ready to uptake the genes.
Yeah, thank you, Aung. May I add some information regarding the transformation in the nature or in the natural condition? So some bacteria has the competence gene called COM, which encodes the protein that helps the uptakes of the extracellular DNA from the environment. There is also pili or pilus specific for helping uptaking the material from the environment. It's different from the sex pili or sex pilus, but this is a specific pilus helping uptaking DNA from the extracellular. Do you get the answer, Nur Awalia?
Yes, very clear. Thank you very much.
Thank you, Arif.
Yeah, thank you for the answer and the question. The next one is Maitza. Please, you can ask your question.
Thank you for the opportunity. My name is Maitza. My ID number 049, and I'm from Microbiology class B. I want to ask about penicillin drug. Penicillin drug can work on bacteria that have a cell wall by inhibiting cell wall formation. But what if the bacteria don't have a cell wall? Because I still don't understand about the mechanism to kill that bacteria. Maybe Ms. Jongkon can explain the mechanism. Thank you.
Okay, wait. Which mechanism that you would like me to explain more?
The penicillin, right, Maitza?
Yes.
Okay. Okay, you mean, yeah? I think normally when the synthesis of the bacterial cell wall, the bacterial cell wall is composed of the sugar we call sugar backbone, which is composed of N-acetylglucosamine and N-acetylmuramic acid, and then they link together, and this sugar backbone, here sugar backbone, and they link together with the kind of amino acid, right? And normally this enzyme, the penicillin binding protein, can link the structure of peptidoglycan, can link the structure of the peptidoglycan. But when the beta-lactam antibiotic, for example, penicillin, go into the cell, this antibiotic bind to penicillin binding protein. And when this protein, actually it is the enzyme, it is the enzyme. And when the drug bind to this enzyme, this enzyme cannot link the structure of the peptidoglycan, cannot build the structure of peptidoglycan. And then when the structure of the peptidoglycan cannot be built, so the synthesis of the cell wall of bacteria is inhibited, is blocked. You imagine that when the bacteria has no cell wall, when the bacteria has no cell wall, so their cell will lysis and the cell death finally. This is the mechanism of beta-lactam, for example, penicillin antibiotic. Do I answer your question, or you can understand more?
Do you get the answer, Maitza? Or do you still don't get the point?
Yes, thank you. But I want to ask again, sir.
Yes, please, you can ask.
This is but what if the bacteria don't have a cell wall? How to kill the bacteria?
In the case of, you mean the bacteria that they don't have cell wall, right? Okay, so you need other kind of antibiotic. I mean other antibiotic that have other mechanism. For example, you need the antibiotic that target the cell membrane, or you need the antibiotic that target the inhibition of DNA or RNA, or the protein synthesis. You got my point. For example, the bacteria in the group of Mycoplasma and Ureaplasma. This is the exception of the bacteria, because as we know that the structure of the bacteria, they have cell wall, but in the case of Mycoplasma and Ureaplasma, they don't have cell wall. So we need other group of antibiotic with other mode of action. Very good question, ka.
Yeah, thank you, Aum, for the answer. Do you get the answer, Maitza?
Yes, already answered. Thank you for the answer, Miss Jongkon.
Yeah, thank you very much. Is there any other or further question for Aum regarding the antibiotics, the mechanism, and also the resistance? So while waiting for the next question, Aum, I have, there is one student who raised the hand. Habib, please, you can ask your question.
Yes, thank you for the opportunity. My name is Habib. I'm from Microbiology Class B, and my student number is 054. And my question is about the process or mechanism the bacterial produce the enzyme. When the bacterial produce the enzyme, is it need time to the bacterial recognize antibiotics and then the produce the enzyme, or several bacterial have their own enzyme to resist specific antibiotics? Can you explain the mechanism more, ma'am? Thank you.
You, Arif, could you please help me about this question? I can get it.
Yeah. Sorry, I was also a bit distracted. Could you please repeat, Habib, your question?
Okay. My question is, when the bacterial produce enzyme, is it need a time to bacteria recognize the antibiotics and then produce the enzyme, or the some bacteria have their own enzyme to resist the antibiotics?
Okay. Do you get the question, Aum?
I cannot understand the word.
So he asked whether the bacteria needs the antibiotic to produce the enzyme to degrade the antibiotics, or the bacteria always produce the enzymes to produce to degrade the antibiotics. Yeah, that's the question, I guess.
I got your point. Actually, to be true, I don't know, but I think maybe they need the antibiotic first and then develop the enzyme. But I'm not sure about this answer. Sorry.
Yeah.
But okay, I got your point.
So may I add some information for some enzyme. The enzyme produced constitutively, so the production or the secretion of the enzyme is always produced. So it doesn't need an antibiotic to be produced, but there are enzyme that needs antibiotics first in the environment, then the bacteria recognize the antibiotic, then the bacteria will produce the enzyme. So this is case by case. You cannot really say that the enzyme that degrade the antibiotics will always produce all of the enzyme, but there is also another enzyme or other groups of enzyme that needs to be induced for the production. Do you get the point, Habib?
Yes, sir. I get the
Thank
point.
you, Arif.
Thank you.
Yeah. Thank you very much. Is there any other question? Yes, Vaishaka, please, you can ask your question.
Okay, thank you, sir and ma'am, for the opportunity. My name is Vaishaka Dandra with ID number 086, and I'm from Microbial class C. So, as already explained, there are broad spectrum antimicrobials such as amoxicillin, tetracycline, and quinolones, which are not specific to only a few types of bacteria. And I have heard and read about using the broad spectrum antibiotics when they are not needed can create antibiotic resistant bacteria. So, what will happen in the future when broad spectrum antibiotics are no longer effective, and what should be done to overcome the more resistant bacteria in the future if the broad spectrum are no longer effective? That's it. Thank you.
Thank you for the question, Vaishaka. Do you get the question, Aum?
No, sorry.
So he asked if in the future the broad spectrum antibiotic is no longer efficient or active against the bacteria, for example, because the emerging of the resistance against broad bacteria, what should be done in order to overcome this problem?
Okay. Actually, there are many ways to solve the antibiotic resistant problem. Even we use the present antibiotics, but use in combination. I mean, if we cannot use only one antibiotic, but we combine the antibiotic together. Maybe we combine the, how can I say, the antibiotic that cannot be used and the antibiotic that cannot be used. I mean the ineffective antibiotic, and we combine together to see the synergistic effect of these combined antibiotics. This is one way. Another way is the can I say, the alternative way, like search for new antibiotics. Search for new antibiotics from many sources. For example, from the natural products, that from plant, from microorganisms, from the marine source, and from animals or something like that. And also the synthetic drug, and use of antibody. It's like monoclonal antibody for the therapy. And what else? Yeah, and also combination between the antibiotic and the natural product. So these are the alternative way to solve the antibiotic resistance problem, I think.
Yeah, thank you for the answer, And the material or the topic about the natural product as antimicrobial agent will be delivered tomorrow, next week.
No, next week.
Yeah. Do you get the answer, Waishaka?
Yes, sir. I got the answer. Thank you, ma'am, from the answer. Thank you very much.
Yeah, thank you. Is there any other questions for Ms. Saising? For Dr. Saising? Maybe while waiting for another question, I have one question regarding the resistance of antibiotics. As we know that maybe some antibiotic has potential to raise spontaneous resistance. Do you know or maybe can you explain the mechanism how the spontaneous resistance of bacteria against antibiotics?
You mean the spontaneous resistance? You mean in the lab or in the?
Yeah, in the lab or in the nature, because the spontaneous resistance can also emerge in the lab and also in the nature, right?
I think so. But you mean the spontaneous resistance? Actually, the bacteria can resist to the antibiotic itself, right?
Yeah.
Do you know about this?
Yeah. So is there any specific mechanism for the emerging spontaneous resistance? Is there any correlation with the mutation maybe?
I think, but for me, if the resistance happen when, I think when the bacteria get the antibiotic, and then they have the like a spontaneous mutation or something like that, as far as I know.
Okay. So that's why it's called spontaneous, right?
Yeah.
Yeah. Okay. Thank you, for the answer. Is there any other question? There is also one question from Puwita. She asked whether is it appropriate to treat the COVID-19 using antibiotic?
As far as I know, I mean, the antibiotic is the agents, are the agents or the substances that used to treat the bacterial infection. I mean, if you need to treat the SARS-CoV or the virus, you need the antiviral. You need antivirals
Okay.
because the target is different, because the virus is not the cell, it's just the particle. So you need the antivirals that target the structure and the process of replication of the virus.
Okay, so the antibiotic itself cannot treat the virus infection,
No,
but the accompanying bacterial infection together with the viral infection, I guess.
Yes.
Okay, thank you, Aum, for the answer. Is there any question? I guess no. So, yeah, do you have any other question from the participants? No, yeah. So I guess because the time is already up, we can also close this meeting for today. Thank you, Aum, for the very nice talk and also the communicative discussion, and also for the participant for the active question and everything. So don't forget for the next week meeting, for the third one, the topic is about the natural product as an antimicrobial agent. Yeah. Thank you again for Dr. Saising for the talk. We will close this meeting. Thank you. Bye. See you next week.
Bye everyone. See you next week.
Thank you. สวัสดีครับ
Bye bye.
Thank you.